Hey everybody! So we are looking at Punnett squares today, not so much this section notes this time. So I figured the best way to do it would be to kind of teach in person here. And so sorry if this looks kind of funky. I'm trying to figure out how to do this kind of live for you. So as always, if you need anything or if this doesn't work for you, then certainly let me know. And if you're a digital learner and at home, we will always go over this stuff on our Zoom calls. So if you need extra help, that would be a great time to ask for that. Okay. So looking here at the worksheet that I use to kind of go over Punnett squares, and this will be posted to Canvas for you. This is the monohybrid squares worksheet, and there are two sets of Punnett squares on this, right? So I can scroll down and see those too. And if you do choose to kind of go ahead and print that, then you have access to that and you can handwrite this with us. Otherwise, it's certainly okay to just draw it if you want. If you're in class, I'll hand this out to you, and so you'll have those with you. This one is the dihybrid Punnett square. Remember, this is where we're crossing two traits at the same time. So we have four alleles, and I'll walk you through that one here in a little bit as well. But again, same thing. It's a single page worksheet. If you want to print it, you'll get both copies there, and you can use this one for practice. I would recommend printing this one because this is quite a bit larger than the monohybrid. Okay, 16 squares to draw is kind of difficult and time consuming. So we're going to start with the monohybrid cross here first. And again, what we talked about with Mendel is that we're looking at one trait, but again, because we're talking about parents and gametes being created from parents, you're going to have two alleles that are potentially going to be passed on, right? We have to look at the probability of inheriting this one or this one since parents are starting with two. So where I usually start with this is to look at some of Mendel's examples here first. So the one we're going to go with, and the one I always go with, is to look at pea plants. And anytime we do any type of Punnett square like this, we always want to identify our alleles first. So we want to identify what's our dominant allele. We want to identify what's a recessive allele, and then what are the letters that we're going to use to represent those. So we'll start with wrinkled versus round, okay? And in pea plants, round is the dominant allele. So usually in genetics, whatever's dominant, that's the letter that we choose to go with. So big R is going to represent the round phenotype. So I'm going to make myself, like a little key up here so I remember what these represent. I would recommend, of course, that you do the same. So big R is round, little r is going to represent the other phenotype, which I just mentioned is going to be the wrinkle trait. And for this, remember, if you keep in mind what we talked about with genotype and phenotype, you have to have a certain combination of these things, right, to see these traits appearing in offspring. So if we start with two purebred plants, okay, let's do a round big R plant and a wrinkled little r plant, and we're going to cross those two parents. So in this case, this is equivalent to what Mendel was doing, right, when he was kind of modifying this process and he was removing pollen from one plant and kind of painting it onto the female reproductive structure, the pistil of the other plant. So when we do this, our gametes are going to be those alleles separating, because in meiosis we know that for this plant, even if this is my genotype, big R, this individual can only give this big R or this big R, right? They cannot give both because Mendel's laws tell us that they separate. So for my gametes, I'm just going to write these down kind of in order. So the first one is big R that he can give, or she, and then big R for the other allele. For this parent over here, they can either give the first little r or the second little r, and so we jot that down there as well. Now, one important thing about this, you want to make sure you keep your parent alleles together, okay? There is a rhyme and a reason to this, and if you don't keep them together, chances are you're going to have some struggles with these Punnett squares, okay? So I usually take this parent and I'll write this parent's alleles on the side axis, and this parent will go across the top. It does not matter where you put them, okay? As long as those parent alleles stay together, then you can put them on either axis. But for simplicity's sake, I generally follow this rule. So I'm going to put a little r above each of those columns, and I'm going to put a big R for each of this other parent's alleles on the left-hand side. So now I've set up my Punnett square. These are my parent alleles, the parent gametes that were created through meiosis, here and here. And then on the interior, remember, this is where we're going to represent our potential offspring genotypes and then figure out from that our phenotypes. So the way that we do that is we're going to take these alleles and we're going to kind of cross them together. So I'm going to bring this one over, big R, and I'm going to bring this one down, little r. Okay, in general, when we're writing these genotypes like this, the big letters are always going to come first. Okay, so that's like your first rule of thumb. So if I have a dominant allele, big R, and a recessive allele, that's going to be my order. Big R is going to come first. For this row here at the bottom, again, I have a big R here, and I have a little r here, and then I have, again, a big R here, and another little r there. So these get passed down just based on kind of dragging these through this Punnett square chart, okay? That's how we write them out. For this one, it's pretty simple. I only have one genotype. I have big R, little r, big R, little r, big R, little r, and big R, little r. So when I talk about genotype, right, these are always going to be the letters that we see in one of these Punnett squares. And so because in this case they're all going to be the same, I can either write all big R, little r, or I could write 100% big R, little r, or I could just simply count them up and say 4 big R, little r. It doesn't matter to me how you do that, but you do want to be able to kind of go from one to the other. So if I have you do a Punnett square on the test, you need to be able to tell me what the percentage is if the answer is written in something like that, or if I ask specifically for a percentage. Offspring phenotypes, remember, this is what they're going to actually look like on the outside, right? It's a physical appearance. And that takes me back up to here. This is where I wrote my little key for this. So I know that big R codes for round and little r creates a wrinkled PC. My phenotypes in this case are based on my genotype. So I know all of my offspring have at least one big R, which tells me, again, based on the rules that we've identified earlier, all you need is one copy of that dominant allele to see the dominant trait. So in this case, all of my offspring are going to be dominant. All of my offspring are going to have that round phenotype. So kind of like I did for the genotypes, I can write this in 3 different ways. I can write all round, 100% round, or 4 round. Okay, and you don't have to write, again, all of those all the time. It helps as we're kind of getting started to see the different ways of representing those, okay? So that's your first Punnett square. Now, I think we need to make it a little bit more complicated because in reality, these things are much more complicated than this. So what we're going to do is we're going to actually take 2 of these, big R, little r's, and we're going to cross them and kind of mimic Mendel's F1s to F2s experiment, okay? So I'm going to go ahead and erase most of this. And I'm going to rewrite up here P's again so that I remember what my key is. And I've got big R again is round. And little r again is going to be wrinkle. So just as a reminder, this is what I'm working with. I told you I'm going to cross 2 of those F1s, those offspring that we just created in that first cross. So that's big R, little r, big R, little r. Okay, so now my parents actually have the same phenotype and genotype, which is not the case, not what we saw in the last one. And in this case, we're going to get a lot of different offspring for the first time. So again, my gametes are just being, written out separately here. So my first parent here can give big R, or they can give the recessive allele, little r. Parent over here on the right, same thing, can give the dominant allele, big R, or the recessive allele, little r. Okay, keep your parent alleles together. And one more time, we're going to set up the axes here. So I'm going to put big R, little r across the top and big R, little r across the side. Okay, if you feel like you're getting the hang of this, go ahead and try to solve this one on your own. You can pause me, all right, and see if you can solve it and then match what I'm going to put up here. If you don't feel like you have the hang of it, then just, walk through it with me again and let's see if we can't figure this one out together, okay? So in this case, I've got big R here, and I'm going to, again, bring them together. So I'm going to get big R, okay? This one over here, I've got big R and little r, and I'm going to bring those together, keeping the dominant allele, again, written first, big R, little r. Here, I'm going to drag these together also, big R, little r. And then for my last one here, bottom right, I've got a little r on the bottom and a little r over here in this column. So it actually is just little r, little r. So for the F2s, which is what these represent, these are the offspring coming from that crossing in F1 and an F1. That F2 or second filial generation, I actually have 1, 2, 3 different genotypes. Okay, do you see how that works? They have different combinations of letters. So again, if I'm going to write out my genotypes, those combinations of letters, I now don't have the ability to just put 100%, right? I have some that are big R, big R. I have some that are big R, little r, and heterozygous. And then I've got just one little guy over here that's homozygous and recessive in this case. So I'm going to kind of do it in that order. I always want to go from most dominant to most recessive. And so when we start writing these, I'm going to start with my homozygous dominant genotypes. I'm going to put 1, big R, big R. And in this case, since I have a bunch of different ones, I'm going to write this as a ratio, which is how we write these things in genetics. A ratio includes a colon, which is like this little symbol right here. Okay, it's not a semicolon, it's just 2 dots, 2 vertical dots, okay? So I'm going to write 1 big R, 2, like compared to, 2 big R, little r, because I have 2 of those right here. And then another pair of dots here, another colon, 1 little r, little r. So that's what we call a genotypic ratio, okay? It helps us to compare all of these items kind of at the same time and side by side by side. There are other ways we can write this. You can write them as a fraction and say 1/4 and 2/4 or 1/2 and 1/4. Or the way that I generally like to do it is to do it in the form of a percentage. And I'm going to write it that way for you too. So 25% big R compared to 50% big R, little r, compared to 25% little r, little r. Okay, same general meaning, right? I have 4 squares either way. It's just here with the percentage that we can apply that to a lot of other scenarios. Offspring phenotypes, remember that's the physical appearance. That's what you see when you actually look at these things. And if we consider the phenotypes of this, well, this becomes a little bit different then, right? I have this one that has 2 big R's. I have this one that has 1 big R, and this one that has 1 big R. And we know from Mendel's work that as long as you have 1 big R, you get that one dominant phenotype. In this case, that's that round phenotype, right? So I have 1, 2, 3 possibilities of creating offspring with that big R round phenotype. So for that, I can count them and I can say 3 round. And then I've got the one over here that's little r and homozygous recessive, which makes its phenotype wrinkled. So 1 wrinkled. Now, just like I did with the genotypes, I can write this as a fraction. I can also write this as a percentage, which is the one I'm going to add right now. So 3/4 is the same as saying 75% are round. And then I've got 25%, the remaining, which are wrinkled. If you do percentages, they do have to add up to 100%. All right, so if you can figure out one percentage, you usually can figure out the next, and so on and so forth, okay? So that's how you write them. It is important that we follow the rules that I mentioned so that the dominant letters are always written first. The dominant phenotypes and genotypes are always written first. That if you're writing a ratio, you're using a colon and not a comma or a slash or a dash or something. Those don't work. That you're keeping the parent gametes together so this parent stays on one axis and the other parent's on the other, and so on, okay? Try your best to keep straight these terms, geno and phenotype. And for me, how I remember it is the way that I've been kind of reiterating it to you, which is to say phenotype, physical appearance, okay? So this is monohybrids, all right? I'm not going to go through another example right now because it's essentially the same thing. There's only so many options we can do with this, but we will practice this more if you're in my class, and we will certainly practice through Zoom with you if you're a digital kid as we need to, okay? I'm going to move on to dihybrids, which are like significantly more complicated, and then we'll be done. So here is my dihybrid Punnett square. Okay, again, it's basically 4 monohybrids together, kind of mushed in one giant Punnett square with 16 squares. The trick with these is we're dealing with 2 genes, 2 different traits, and each of those is going to have 2 alleles. So you're dealing with a total of 4 alleles coming from potentially each of two parents. And so you have to look at the combinations of those because, again, these things assort independently. So if we stick with P's, because, why not? We could keep the big R and the little r stuff that we've already been working with, right? Because that's simple enough. We can write big R is round, and little r is, again, going to be wrinkled. And then in addition to that, I'm going to go ahead and use P color in this case. Okay, so we're going to do big Y, which is actually the dominant form, and that's going to be yellow. And then little y, which is going to be green. Now, one thing that I did here that's going to be super important that you probably haven't thought of, and I got a big glare up there. I'm not going to be able to fix it very well, but what you need to notice about this is the way that I wrote the y's over here, and I can pick this up and at least show it to you for right now. So notice my y's look very different from each other. The reason for that is because I need to be able to easily distinguish between those two when I go and actually write out my genotypes to decipher my phenotypes. If you write your R's like I normally do, which is like just this, like my capital and lowercase y's look very similar. I know one is one like when I'm writing it in a word or when I'm starting a word, but for the most part, I never write this little loop at the end of my Y, okay? Now, I do that always when I'm doing genetics problems like this because I want to make sure it's real clear to my teacher that I know what I'm doing, and it's also real clear to me when I got to write out this stuff later that becomes super complicated that I know what I'm doing, okay? So make your letters look different, all right? Whether it has a line above it, a line below it, one's in bold, one's in a different color, however you want to handle it, but make sure that they appear to be very different, okay? So my parent genotypes, we're going to start with a very easy example, and then we're going to do the hardest examples. We're going to do both in this case. Parent genotypes for this. I'm going to start with one parent who's homozygous dominant for both traits. All right, and I'm going to rewrite this Y because it looks kind of funky. Homozygous dominant for both traits. So if we're thinking about these two traits, if it's dominant for both, that means it's going to be round and yellow. Okay, I'm going to cross that homozygous dominant parent with a homozygous recessive parent, little r, little r, little y, little y. And the only reason why I'm putting r first is because I wrote it first. Okay, but notice how I'm writing these. The r's are going to stay together and the y's are going to stay together. They're never going to separate throughout this whole thing. And I'll show you what that's going to mean in a few minutes. Gametes. There's a mistake on this worksheet. You need to add 4 additional lines here. Those 4 lines are going to represent the parent over here. Okay, the first 4 lines are the 4 possible allele combinations that we can get from parent 1 right here. Okay, so I realize that parent 1, the 2 big R's look exactly the same, right? This is, again, your easiest example. All right, and so for parent 1, again, they're going to give 1 R and 1 Y. Because they all look the same, parent 1 is able to give, let's say, this big R and this big Y, but this parent can also give this big R and this big Y. And this parent can also give this big R and this big Y, and this big R and this big Y. So there's actually really 4 combinations, even though for all intents and purposes right now, they're going to look the same, okay? But we need 4 lines because we have 4 rows and 4 columns. So we're going to write the first parent's gametes up here, big R, big Y, big R, big Y, big R, big Y, big R, big Y. The parent here, second parent, which is homozygous recessive for both, little r, little y, little r, little y, little r, little y, little r, little y. Okay, so we've got all our gametes. Again, your parent gametes need to be kind of in the same place. They need to be on the same axis. So my first parent here, I'm going to write across the top this time, big R, big Y, big R, big Y, big R, big Y, and big R, big Y. Okay. Second parent here, I'm going to go ahead and put on the side. Again, it does not matter which parent is where. Second parent can also go across the top if you wanted. You can just swap them. You'll get the same information in the inside. Okay. So here's the catch. We're going to start pulling these all together, but like I already said, the R's are going to stay together and the R's are going to stay first because I started writing them first. So my offspring genotype in this first box is actually going to be big R, big Y, little y. weird because I just got done telling you in a monohybrid that the dominant letters are always going to be written first, and here I'm not doing that. Okay, and the reason for that is this rule of keeping similar alleles together is going to trump anything like the dominance rule that I told you about earlier. So it's more important, in other words, that we keep the R's together than it is to write this dominant Y earlier. Okay, so the R's always stay together, the Y's always stay together. All right, keep that in mind. That's a really good rule of thumb for this. If I continue here, I've got a big R, a little r, a big Y, and a little y again. Big R, little r, big Y, little y. If I continue doing that, you can start to see what's going to happen here. I've got a lot of the same genotypes appearing because, again, my parent gametes are exactly the same. So it doesn't matter if it's this little r and this big RY, if it's this big R and Y and this little R and Y, I'm always going to get the same result, right? Because I'm consistent in how I'm writing them. So what that means for this is you don't have to write it 16 times. I do not want you to write it 16 times. If you were in my class doing this with me, we would not write it 16 times, okay? I would do one of these. And on a test, if I asked you to draw this, I would say go ahead and do that too, because what that tells me is that you know that this is just repeating in all 16 squares. If you want to write it 16 times, go ahead and write it 16 times. It's going to take up more time than it's worth, okay? But this is what we end up getting here as a result, okay? Now, that means all of my genotypes are exactly the same. So just like we did in monohybrids, I can write these some different ways. I can say all are big R, little y. I can write 100% R, big R, little y, little y. I can count and say all 16 are big R, little r, and big Y, little y, okay? Any of those would be just fine. Again, I like to talk about percentages more, but in dihybrids, it becomes a little bit more complicated to do that, okay? So genotypes, again, are just those letters. Phenotypes, if we think about what these things look like, right? In this case, I'm going to have to write two characteristics down. So it's not just round and yellow. It's going to have to be, you know, round or green, and both have to go together in this case, all right? So my offspring phenotypes, all of them have at least one big R, and all of these have at least one big Y. So that tells me that all of my offspring are going to be round and yellow, because big R specifies the round phenotype. Big Y tells me it's got to be yellow in color. Okay, again, I can write it as 100% round and yellow, or I could write it as 16 round and yellow. Okay, so that's how we set one of these things up. Now, again, I mentioned I'm going to complicate things for you, and we're going to do the toughest one now. Okay, so if you felt like that was easy, that's a great sign. Like, we can move on to the next thing. But if you're not quite there yet, you know, maybe reverse and listen again and watch again and see if, you know, you can't get a little bit more comfortable with how we're setting these up. All right, so again, what I'm going to do here for this next one is we're going to keep the same traits. All right, and this time I'm going to try to just erase part of this because I don't want to have to rewrite what's up at the top there so much. So our offspring, again, from this cross that we just saw, represent what we call our F1 generation. In that F1 generation, we're going to take two of those this time and cross them, just like we did in the monohybrid. We want to see what happens if two of these F1s are crossed. And so that means my two genotypes that I'm going to start with here as parents are going to be big R, little r, and big Y, little y. All right, so let's see. Go ahead and write that down here. So we're going to start with, again, big R, big Y, little y for both. And probably kind of immediately you can see how this is going to be more difficult because I have some different alleles showing this time. My gametes, again, I want to add my 4 lines for the parent on the right. But in this case, my parent gametes are going to actually be kind of exactly the same because my parents are exactly the same. However, within a parent, I'm going to get 4 different types. Okay, so what I mean by that is I'm going to compare and try and catch all of the possible combinations of R's and Y's in this parent and then do the same for this parent. The way that we do that is in a method called FOIL. And if you're in algebra and you've started FOIL, which usually is around this time, FOIL is a process that you use to help solve the equations, right? And it's order of operations. So when we talk about FOIL, it's first, outer, inner, and last. That's what it stands for. So FOIL, first, outer, inner, last. What we're doing with that here is we're going to do that to identify our gametes. And if we do that in a specific order, we're going to get a really cool little pattern here in our Punnett square. It just helps you to know that you're doing it the right way. So I'm going to take my first R and my first Y. That's going to be my first gamete. So the F in FOIL, first R, first Y, gives me big R, big Y. Okay. Now the O in FOIL is my next letter, and that means I'm going to pick my two outermost letters on this parent right here. So the two outermost letters are this big R and this little y right here. Okay, so I'm going to write big R, little y. After F and O in FOIL, I have an I, and that I represents the two innermost letters, which is my little r and my big Y. And then I have my last R and my last Y. That's my L in FOIL. So little r, little y. Okay? Notice each of those four gametes is different. They are not the same. So it's important that we match them up properly to ensure that we don't miss any possible combinations of R's and Y's. Same with this parent over here. Because those parents are the same, I can essentially copy those over. But if you want to practice that FOIL technique again, by all means, do the same over here and see if you don't get the same answers as what I just wrote there. Okay. All right, so parent alleles again, parent gametes are going to stay together. I'm going to write them in the order that I wrote them down using FOIL. Again, you don't have to do it this way, but if you do, you're going to get a really nice pattern, okay? And it's a good way of ensuring that you're doing the right thing. So those are my gametes. Notice, again, my R's are still in front of my Y's. It looks weird, and it is weird because it violates that other rule we talked about, but I need to keep my R's first, okay? So writing out my genotypes, again, I'm going to keep R's together and in front of the Y's, and then within those R's and Y's, I'm going to write the dominant letters first. So this one is going to be big R, big Y, big Y. This one here, I'm going to write big R, big Y, little y. This offspring, big R, little r, big Y, big Y. This one over here on the top right, big R, big Y, little y. Okay, if you saw how I did that and you feel like that was what you would have gotten, again, pause the video, see if you can't do it on your own, and then come back and check your work and see if it's going to match mine. So I'm going to go through the rest of them right now. Here we've got big R, big Y, little y. Next is big R, little y. Here I've got big R, big Y, little y. Big R, little y, little y. Next row, big R, big Y, big Y. Big R, big Y, little y. Little r, big Y. Again, one of those weird kind of looking genotypes. Little r, big Y, little y. Big R, big Y, little y. Big R, little Y, little y. Little r, big Y, little y. And the last, never least, little r, little y, little y. All right, so 16. And if you're staring at that like students normally do in my class, they're like, what the heck did we just do? Like, that's crazy. It doesn't mean anything, right? Well, it does actually mean something. What it can tell us is these two things. Again, our genotypes and our phenotypes, the likelihood of inheriting both round and yellow, or wrinkled and yellow, or round and green, or wrinkled and green. And that's what we're going to try to identify. The way that I like to do this to simplify it, because if we go through and count each of these genotypes separately, like I only have one of these, and I have one of these, and I've got like 4 of these in the middle, but beyond that, like it's complicated. There's a whole bunch of different ones. So what I do to make the process of going from genotype to phenotype easier, and what I want you to do also, is to look at these genotypes and identify first how many will have a specific phenotype. So if we go from the most dominant pairing, right? If we say, hey, I want to find out how many of these offspring genotypes will create plants that have round and yellow pea seeds, that means we're going to look for, in our Punnett square, how many have at least one big R and at least one big Y. Because again, it doesn't matter if it's big R, big R, big Y, or if it's big R, little y, they're still going to look the same, right? Physically, that phenotype, that physical appearance, is still going to be round, it's still going to be yellow. So go through and count and see how many we can come up with that are both round and yellow, meaning they have at least one big R and one big Y. And I'm going to do the same right here. Okay, I'm going to use a highlighter for mine. If you're doing this on paper, it'd be cool to use a highlighter so that way you can still kind of see what you've done, but you don't over count something. So what you end up with here is going to be a pattern that looks a little like a triangle of sorts. All right, so I end up with all of these here. 1, 2, 3, 4, 5, 6, 7, 8, 9 that have a big R and have a big Y. And that's going to tell me that I'm going to have 9 that are round and yellow. How I write out that genotype, instead of trying to divide 9 by 16 and get a percentage, I just write 9. So I'm going to say I have 9 of my offspring, 9 of my potential possible offspring, I should say, that have a big R. And then I'm going to put a little like underscore there, like a little line. And that's going to represent that it's kind of like a placeholder. In other words, I don't care if it's big R or if it's big R little r. This is going to show me, though, that I do know that there's another allele there. Okay, so 9 big R with a space, big Y with a space also. Okay, and I can go ahead and write out my phenotype, right? It's 9 round and yellow. All right, now remember when I write a ratio, I want to keep those symbols, those colons in between. So I'm going to go ahead and put my 2 dots. Same here, my colon, I need my 2 dots there. And the next one I'm going to count is the next dominant. So let's say, because R's are first, let's say with round, I want to know how many are round and green. Okay, so how many have at least one big R, but have 2 little Y's now? All right, and I'm going to go through and highlight the ones that I see. 1, 2, 3, and that's it. And notice I've already taken out like a huge chunk of this, just looking at those 2 different types. So I've got 3 of them, and I'm going to write it very similarly. I'm going to put 3 big R with a placeholder again, because I have big R, I also have big R, little r. And then I'm going to put 2 little y's this time, because I know to have a recessive trait show, I have to have 2 recessive alleles in the genotype. So I go ahead and put 2 little y's there in that case to represent that I know that they have to be homozygous recessive. If I do my phenotype, 3 round, and then the recessive is going to be that they are green in color. And then I'm going to again put my colons for the next grouping. All right, I only have 2 more options left. So let's again do the most dominant ones that are left. So most dominant would be to say wrinkled and yellow. So I want to find how many have at least 1 little r, or I should say 2 little r's this time because it's recessive, and at least 1 big Y. All right, so if we look at that, they all fall in the same kind of general area, right? I don't have much of anything really left in this Punnett square, but any of these that have at least 1 big Y left will all fit in this category. And so that's 3 of them. I have 3 of those. So I'm going to put 3 little r, big Y with a little placeholder. Again, to show that I know that they have to have 2 recessive alleles to show that recessive trait, but they can have more than 1 dominant allele, or they can just have 1 to see the dominant yellow phenotype. So my phenotype for this, I can go ahead and write 3 wrinkled and yellow. All right, and then I only have 1 more little lonely, only homozygous recessive for both traits individual right there. Okay, so for this one, I'm just going to go ahead and write out the whole genotype. 1 little r, little y, and then again, 1 wrinkled and green down here for my phenotype. Okay, so that's kind of how you set up a dihybrid like that. You will see more examples. I'm going to ask you to do some of these, likely for homework or in class if you're in class with me. And so make sure, if we struggle on this, go back, replay it, go back and look at the FOIL method if that's something that, seems like it's new to you. Look at how we counted these things up. And if you've got questions on any of this, as always, please let me know, because you will see Punnett squares like this on your test. So you do need to feel comfortable with them. Okay. All right. That's it for right now. If you need anything else from me, let me know. Otherwise, I will let you go. Bye, y'all.